Hello, this is the 20th lecture in the series of Principles in Biochemistry from the book edited and written by Albert Lehninger, Principles of Biochemistry, 7th edition. Today we're going to wrap up the discussion of membranes and transport mechanisms and the bioenergetics with a discussion of tyrosine kinases and receptors. As we discussed, a lot of these receptors are coupled to G protein, which helps ameliorate the transport of substances and involved in the second messenger production. Well, there's also a different function of G protein, and this is a different mechanism from the G protein coupled receptors. We're talking about tyrosine kinases. So in this example, we have a ligand binding to the external membrane, and there is a protein kinase on the cytosolic side of the which phosphorylates tyrosine. So kinases, by definition, are enzymes which phosphorylate substances. So tyrosine is phosphorylated. Prototypes for this are insulin and epidermal growth factor. So for insulin, protein kinase phosphorylates tyrosine of selected proteins, and these target a variety of domains. One broad category are RAS. This is a family of G proteins. They will utilize free GTP, and they are utilized for free GPT for bound GDP, and they modulate or mediate numerous biological functions as a cascade. So one example of this is membrane phospholipid PIP3, and this is another target of cyclic AMP, and this will leads to protein kinase activation and reduced glycogen synthesis. So the combination of inactivation and activation pathways amplifies the insulin signal. We also have crosstalk where the signaling systems are integrated with each other. So one example is receptor guanylyl cyclase, where GDP or GTP is converted to cyclic GMP, and this acts on multiple organs. So in the kidney, you have atrial natriuretic factor, and that is released to stretch cardiac muscle. It's a laboratory analyte that's is correlated to stretch cardiac muscle, which is a component of congestive heart failure. You can also convert GTP and cyclic GMP in the kidney, and you can get increased renal excretion of sodium and water, lowering the blood pressure. In the intestine, cyclic GMP increases chloride secretion and decreases water reabsorption, so you get watery stool under this circumstance. The signaling proteins can be multivalent. They can interact with multiple binding proteins simultaneously. There are scaffold proteins. They can bind with multiple kinases. The phosphatases can remove phosphate from serine and threonine and reverse the pathways that were affected by the protein kinases. Also, there are membrane rafts, which are aggregates of lipid receptors. They're embedded in the fluid membrane matrix, and they facilitate complex binding behavior. So you can actually have clusters of recognition sites with what are called membrane rafts, which are lipid receptors aggregated on the surface and moving about the membrane. Gated ion channels are really critical to much of this. We have excitable cells. This is where the messenger is the change of membrane potential. You couple this to an ion transport, such as a sodium-potassium ATPase, and this creates a charge imbalance because the system allows to have 3 sodium atoms or ions out and 2 potassium ions in. And the calcium concentration is too low to have any impact on the transmembrane charge. So the membrane potential depends on composition of various ion channel openings, and these will determine the neural-induced contractions and hormone secretions. And when you look at the imbalance of the ion movement, you get a cell interior that's about -60 millivolts relative to the outside. So they're voltage gated ion channels with sodium channels open and depolarized. There's efflux of potassium ion. Then there's an open calcium channel which triggers acetylcholine, and it carries a signal to another neuron. So when we look at this rigorously, we have a voltage gated sodium channel here with sodium coming in. There is in the axon, the presynaptic neuron here. You get potassium moving out for repolarization. So depolarization, sodium's in, potassium comes out, so it's repolarized, and then you have the calcium being brought in also at this point through a gated calcium channel. There are vesicles which contain acetylcholine, and the calcium helps with the movement of the acetylcholine across the synapse to the receptors. So this is a presynaptic neuron, postsynaptic neuron, and the acetylcholine is recognized and brought aboard by acetylcholine receptor ion channels, and the action potential is propagated through neurons using this mechanism. Again, initiated with calcium influx, potassium efflux, secretory vessels involving calcium brought in, and this facilitates movement, and the signal transmits through subsequent nerves. So the unidirectional wave of depolarization extends the length of the axon. Acetylcholine is its own messenger. These are extracellular ligands. The second messengers are intracellular. The ion channel blockers are seen in some animal neurotoxins. They're heavily amplified. There are also nuclear hormone receptors which act directly in the nucleus. They're highly hydrophobic. They rely on carrier proteins. They target tissue, and they have simple diffusion across the membrane, and they alter gene expression through binding with the hormone response elements. So when you see this, you have a hormone right here bound to a serum binding protein. The bound hormone is there, and then the hormone is brought across the nuclear membrane. So this is the receptor brings the hormone across the plasma membrane through the cytosol, and then is brought across the nuclear membrane. There's a nuclear receptor here which attaches here, and this amplifies the effects of RNA polymerase. You get increased amounts of transcription from the DNA template to messenger RNA, and then translation of the messenger RNA in the cytosol with ribosomes, and then increased amounts of protein that are produced with altered cell function. The prototype for this type of would be oftentimes the sex hormones, estrogen and testosterone, or among the ones under consideration. These effects are not immediate. The binding causes delayed metabolic changes. There's also biosignaling in microorganisms and plants. E. coli membrane can detect extracellular nutrients. Flagella propel these nutrients. There are two signals. One is a histidine kinase, and then there's a response regulator, and the flagella moves to increase the concentration gradient. Plants also possess a large variety of protein kinases, but there's no G protein clustered receptors nor cyclic nucleotide activated protein kinases. These are absent from plants. The cell cycle in protein kinases, in the S phase, you get replication of DNA. In the G2 phase, under mitosis, the new proteins are synthesized. The cell doubles in size. The M phase is where the paired chromosomes separate with the envelope breaking down. A G1 phase is the resting phase where the RNA and protein synthesis occur. and G0 is a quiescent phase with a variable time frame. Protein kinases are involved in mitosis. They're factors in all phases at multiple sites, and the phosphorylation of threonine initiates the cell cycle. Cyclins are broken down in certain cell phases. These are facilitated by specific proteins that are tightly controlled during mitosis. There are growth factors and cytokines. These also stimulate protein kinases and cyclin. There's a membrane breakdown protein called lannin. This is in the meshwork of the membrane. It is phosphorylated by protein kinase, then depolymerizes. There's additionally an actin myosin contraction, and that's inhibited by a similar mechanism that you would see with lannin. For oncogenes, neoplasia is by definition an uncontrolled growth of tissue in a biological system. You can't inherit this. Broadly speaking, about maybe a third have inherited instances. About a third of them are environmental, and then the final third can be multifactorial or have no specific known cause. So you've got inherited, environmental, and genetic damage, i.e. mutations, from whether they're chemically formed or radiologically formed, or if they're just simple errors in the cell cycle and they cannot be repaired. The oncogene is a mutated version of the gene used in or involved in cell regulation. We also talk about proto-oncogenes. Proto-oncogenes are found in the animal hosts. They're coded by viral carriers into host genome. Oncogenes are genetically dominant and a variety of products that facilitate growth or produce. So oftentimes there's the receptor tyrosine kinase in neoplastic conditions unregulated, so you get uncontrolled and rapid growth of tissue with no inhibition mechanism available. Among mutant forms, the RAS oncogene codes a protein that binds GTP but cannot be degraded, so that gene is always on. And then there are tumor receptor genes. These are involved with restraint of cell division. There are mutations in which lead to tumor formation. There's a recessive condition, for example. Both genes need to be mutated for the recessive condition. One example of this is retinoblastoma, which is an eye tumor of childhood, and these are defects of the RB gene. There are stability genes. Stability genes are involved in repair. The BRCA1 oncogene is involved in breast and ovarian cancer, and you see P53 involved in squamous cell cancer. Sometimes a series of mutations is required, so colorectal cancer is an example where you have to have multiple hits or multiple insults to the genome before you get an irreversible neoplastic scenario involved, and there's much characterization of colorectal cancer involving broad classifications of these mutations. There's also driver mutations. Driver mutations are single mutations, and by themselves they cause unregulated cell division, affect cell survival, preservation of genome, and mechanism of cell action. So these are one hit as opposed to the multiple hit model. And apoptosis is cell death, and this is usually programmed, and you see these in cells with DNA damage, autoimmune disease, menstruation, and autumn dropping of leaves is an example of apoptosis. Apoptosis has a particular look under light microscopy, and oftentimes they occur as isolated cell death that you can spot early on in a process, as oftentimes a first form of a neoplastic process or other condition that's going on. So we talked about bioenergetics and metabolism. We talked about this in the first talk, where you have obtaining chemical energy. Nutrient molecules are converted into cellular constituents. They polymerize macromolecules. synthesize and degrade biomolecules. Autotrophs, again, use carbon dioxide, heterotrophs, or hydrocarbons, and they depend on autotrophs for the carbohydrate. And nitrogen is where ammonia and nitrate are used for bacteria in plants, and amino acids for animals. And this will be part and parcel of some subsequent talks that we'll be doing that employ this particular issue. That'll be it for this talk. Thank you very much.